Hello, today we will be presenting on the single nucleotide polymorphism associated with RS2736990 and the increased risk of Parkinson's disease, as well as other phenotypic traits. This presentation is created and presented by Group 3, consisting of Joanne Rubalcaba, Saneh Kahlon, and Sandra Carranza.
So to start off, what is SNP? SNP is a single nucleotide polymorphism. It's a common type of point mutation among individuals. Each SNP represents a single DNA building block, such as a nucleotide or base pair that has an experienced modification. These can create changes that are synonyms, that does not change the amino acid coded for the DNA sequence, or they can be non-synonyms. When the amino acid encoded for the DNA sequence is changed. So the goal of our project was to analyze and interpret anonymous ancestry testing data that includes the chromosome, the location, the RS number, and the genotypes in order to make predictions of the phenotypic traits and disease risks. Our second goal was also to determine how SNPs can influence health and or disease, particularly Parkinson's disease.
All right, so let's begin with an introduction to Parkinson's disease, often abbreviated as PD for short. So Parkinson's disease is a neurodegenerative disorder which is usually identified by the gradual degradation and eventual loss of dopamine-producing neurons in the brain. And this is pictured here on the left, where we see a comparison of a normal brain to a brain with Parkinson's disease and how neurons often degrade and are eventually lost. At this point, Lewy bodies often become visible, which are abnormal accumulations of protein in the SN brain region. Now, genetically, PD can have different subgroups. If we look at PD with autosomal dominant inheritance, we find that mutations of the PARC1 and PARC4 genes are usually involved. In contrast, if we look at PD with autosomal recessive inheritance with early onset systems, we find that mutations of the PARC2 and PARC6 genes are often involved. Now, outside of just these loci, it's also possible that trinucleotide repeats in other genes and other loci regions can also contribute to a diagnosis of PD. Familial history and environmental factors can also contribute to the progression of this disease. Now, when we examine the single nucleotide polymorphism associated with RS2736990 in particular, we find that it's located on the synuclein alpha locus of chromosome 4q22, which is often abbreviated as SNCA for short. Now, this SNCA gene is often located in presynaptic terminals of neurons in dopamine-producing regions of the brain and is thought to maintain a healthy number of synaptic vesicles. This gene also encodes for the alpha-synuclein protein, which is very abundant in the brain. Mostly, it occupies nerve terminals in order to control synaptic transmission and the healthy release and inhibition of neurotransmitters. So when there's mutations in the SNCA, as is represented by the single nucleotide polymorphism associated with RS2736990, it's thought that misfolded alpha-synuclein proteins are produced in abundance. And what happens is that these misfolded proteins aggregate to form Lewy bodies, which disrupt normal neuron function and cause eventual degradation of neurons and brain activity. So when this particular single nucleotide polymorphism, SNP for short, was examined in a genome-wide association study, it was found that a majority of the healthy controls possess the T allele, which is thought to be the major allele at this location. However, missense mutations increase the frequency of the minor C allele by replacing the T alleles characteristic of the healthy controls at this very location, and were thought to contribute to the increased risk for Parkinson's disease that is associated with this SNP.
So there were several studies that have examined the relationship between RS number and Parkinson's disease. So to start off, they first received an anonymous ancestry data set, which included the RS number, the chromosome, the position, and genotype. Second, they identified one human genetic disease, which was Parkinson's disease caused by the SNP via web-based database, which was named OMIM. There they located the RS number in the web base SNP media database to determine associated phenotype genotype relationship. They then analyzed Ancestry data set to locate the RS number and determine the genotype of the anonymous donor at the SNP. Finally, they analyzed the anonymous donor's genotype via comparison of the genotype-phenotype relationship data present on the web-based OMIM and the SNP media bases to predict the susceptibility to the risk of developing Parkinson's disease and other phenotypic traits.
Upon completion of our methods, we achieved our data and results. One of our results is this Ancestry distribution table of the CC, CT, and TT genotypes observed amongst the Ancestry samples from the SNP EDIA database for the SNP identified by RS2736990. Also from the SNP EDIA database, we obtained this phenotype-genotype relationship table for our analyzed SNP, which depicts the CC, CT, and TT genotypes and their associated phenotypes of increased risk for PD. So these are the results obtained from locating the SNP in the provided Ancestry dataset. For interpretation purposes, it is important to note that while the anonymous donor has a reported genotype of AA for the SNP identified by RS2736990, base pairing rules inform us that a genotype of AA correlates with the presence of TT, which is used for analysis and comparison with the results that we obtained from SNP EDIA in the earlier slides. So in order to interpret and analyze the genotype data of the anonymous donor to predict whether they are at increased risk of developing Parkinson's disease, we compared the Ancestry data to our results from the phenotype-genotype relationship table on SNP EDIA for the SNP associated with RS2736990. Now we find that anonymous donor depicts a AA genotype as demonstrated by dataset 2. This corresponds to the genotype of TT with a magnitude of 1 as demonstrated by the phenotype-genotype relationship table. Thus, based on their genotype, the anonymous donor has a normal risk of developing Parkinson's disease. So in order to interpret and analyze the genotype data of the anonymous donor to predict their ancestry, we then compared the Ancestry dataset to our results from the Ancestry distribution table of the CC, CT, and TT genotypes obtained from SNP media for the SNP associated with RS2736990. We know that the anonymous donor depicts a AA genotype, as was demonstrated by dataset 2. Now, Ancestry data indicates that amongst the available ancestries on SNP EDIA for this SNP, those of Tuscany ancestry and Italia have the highest prevalence of the corresponding TT genotype for the AA genotype, with a prevalence percentage of 31.7%. Thus, based on their genotype, the anonymous donor is predicted to be likely of Tuscany ancestry from Italia. And finally, in order to interpret and analyze the genotype data of the anonymous donor to predict whether they are male or female, we analyzed dataset 2, which is where we obtained the genotyping and ancestry data for the anonymous donor. Now, the anonymous donor depicts genotyping results on several locations on both the X and Y chromosomes, as is demonstrated by dataset 2, which is pictured on the right. Now, the Y chromosome is only present in males, while the female possesses only 2 X chromosomes. Thus, based on ancestry data and the idea that genotyping results are available for both the X and Y chromosomes for this anonymous donor, the anonymous donor is likely predicted to be male. So as a result of this project, we learned how to predict phenotypes and traits with the presence of genotype data. By comparing genotypic data of an anonymous donor at a single location to the comprehensive data that is available for that particular single nucleotide polymorphism on web-based databases and primary research literature, we learned how to predict traits such as disease risk, ancestry, and whether an individual is male or female. We believe these skills are invaluable and helpful because they allow us to understand the genotype-phenotype relationship and conduct similar analyses for family, friends, and ourselves when presented with similar data in order to gauge important aspects of our own health, such as disease risk, and aspects of our identity, such as ancestry. Thank you so much for listening to our presentation. Here is our author's contribution statement and our works cited. Thank you.